Okay, so welcome back from the break. Let's take a look at the concept check questions very quickly, and then let's talk about translation. So if there are only about 21,000 genes in the genome, how is it that we can make about 75 to 100,000 proteins? And the answer is obviously because of the presence of the introns and the exons and the ability to be able to splice together those exons in a number of different combinations to produce multiple messenger RNA molecules from a single gene on the basis of how those exons are spliced together. So that's how we make all of those proteins. If a TV show is an analogy to a eukaryote transcript, what would the introns be? The introns would obviously be the commercials in the middle of the program. So if you wanted to see the program in full without the commercials, you would have to cut them out and splice together the sections of the program. So the commercials would be the equivalent of the introns. What effect would removing the GCAP have on a messenger RNA? Well, remember the GCAP there is to do three things. Number one, it allows for the export of the final messenger RNA from the nucleus, so that wouldn't happen without the GCAP. The GCAP is also there to stop the enzymes in the cytoplasm from immediately chewing up the messenger RNA. It has a protective role. So again, if it was missing, that would happen, it would get chewed up. And remember also the GCAP identifies the 5' end of the messenger RNA to the ribosomes. So in the absence of the GCAP, the ribosomes wouldn't know which end was the 5' end, and they wouldn't attach correctly to the messenger RNA, and therefore translation would get totally screwed up. So that's the answers to these concept check questions. Let's go on now and talk about translation. So when we start translation, there are a number of players that are involved. You've already met one, the messenger RNA, but there are two others that are involved. The next one that we want to talk about very quickly is the transfer RNA or tRNA. This is the cellular interpreter because it speaks both nucleotides, because it is an RNA molecule and therefore it's a polynucleotide, and it also speaks amino acids. So let's take a look at the artist's rendition so that I can show you how it does that. The text tells you here, but it's a little bit easier to show you on the basis of the diagram. So here is a two-dimensional diagram, and it shows what the RNA molecule or the tRNA molecules look like post-translation, because they are the product of translation. They are, after all, an RNA molecule. Because there is a certain degree of base complementarity, base pairs form, and that causes the tRNA to fold into this structure called a hairpin loop structure. It's very characteristic of the tRNAs. So this is called the hairpin loop structure. There are two specific regions that are critical for the function of a tRNA. There's this set of 3 nucleotides in the middle loop of the 3-loop structure called an anticodon. And the reason why it's called an anticodon is because it actually base pairs to a codon in the messenger RNA. So we can figure out what the codon would be that this could attach to. So A complements U. So an A complements U, and G complements C. So this would be the codon sequence in the messenger RNA that this anticodon would attach to. Now, why that's important is because at the other end of the tRNA, there's this sequence of nucleotides to which an amino acid is going to be attached by an enzyme that recognizes the particular amino acid and attaches it only to a tRNA that has the anticodon that would bind to the codon for that amino acid. So UUC happens to be the other codon for phenylalanine. So at this end, you're going to get a phenylalanine amino acid attached to this. So this is why a tRNA can act as the interpreter between the language of nucleotides and the language of amino acids. It speaks nucleotides because it is an RNA molecule, and specifically it's the anticodon that is involved in the interpreting. Remember, it binds to a codon in the messenger RNA. And why that's important is because at the other end of the tRNA, the amino acid that is coded for by the codon that the anticodon attaches to is going to be attached to the amino acid binding region. So that's how these tRNAs carry out the interpretation of the codon sequence in the messenger RNA and turn that into an amino acid sequence by understanding the codon in the messenger RNA and bringing the appropriate amino acid for that codon. So how does the particular amino acid get attached? Well, there are enzymes called amino acyl tRNA synthetases, you can see that here, that recognize a particular amino acid and the tRNA to which it needs to be attached, and will only attach the particular amino acid that should be attached to that particular tRNA. So this also guarantees the right amino acid being brought in by the right tRNA. Remember, the tRNA will only bind to the appropriate codon for the amino acid that it carries. The right amino acid is guaranteed by the enzyme only recognizing the particular amino acid that should be attached to that tRNA. So through this two-step process, guaranteeing the right amino acid is attached to this tRNA and that this tRNA only binds to the codon that codes for this tRNA, you can guarantee that the right sequence of amino acids is assembled into the polypeptide. Now, you might think that because there are 61 codons coding for amino acids, remember 3 of the codons are stop codons, and yes, there is a start codon, but it also codes for methionine, the amino acid. So there are 61 amino acid coding codons that you might need 61 tRNAs, but you don't. In fact, you need substantially less. And the reason why is because of that wobble base situation. Remember that for many of the codons where there is redundancy for the amino acids, the difference is in the third nucleotide, otherwise known as the wobble base. As long as the first 2 are correct, you're going to get the right amino acid, which means that the tRNA actually only needs to recognize the first 2 nucleotides. Doesn't matter what the third one is. So long as the tRNA can recognize the first 2 nucleotides and attach to those, it's going to bring the right amino acid. So it turns out that in fact, multiple codons can be serviced by a single tRNA because of the wobble base situation. So in fact, there are substantially less than 61 tRNAs. I believe the number is somewhere around 40 to 45 is the accepted number at this point. It may actually be less than that, depending on what the feeling is. I haven't seen the latest information on how many tRNAs we actually now recognize are necessary, but the last number that I saw was somewhere around 40 to 45, but substantially less than the number of actual codons. The other major player is the ribosomes, the actual complex of proteins and 2 RNA molecules known as rRNA that do the assembly. So the proteins in the rRNAs are assembled into 2 complexes or subunits known as the large subunit and the small subunit. They work together to clamp the messenger RNA between the 2 subunits. The small subunit acts as a regulator and helps identify where the start codon is and therefore guides the large subunit to clamp around the messenger RNA in the appropriate position with respect to the start codon. That then establishes the reading frame. Remember, that's how we frame out the nucleotides into groups of 3 as codons, and allows the ribosome then to read off the sequence of codons and assemble the polypeptide according to the codons. Within the large subunit are the enzymatic activities. And here again, we run into an RNA molecule serving as an enzyme, because the rRNA molecule in the large subunit is actually the enzyme, a ribozyme, that assembles the amino acids together by forming peptide bonds. The protein complex then of the large subunit forms 3 sites that assist in the process. So the sites are known as E, P, and A. E is real easy, E for exit. The tRNAs, having completed their role in translation, leave through the E site. The P site, the textbook refers to as the peptidyl tRNA binding site. Think P for polypeptide. A lot easier to remember, because the tRNA in the P site is always going to have the polypeptide attached to it, the growing polypeptide attached to it. The A site is the location in which the tRNA bringing the next amino acid is going to locate itself. So at the base of the A site is going to be the next codon in the sequence. And so the A site, the book refers to as the aminoacyl tRNA binding site. But just think A for amino acid, because the A site is always going to contain the tRNA that brings with it the next amino acid to be added to the polypeptide. So here you can see the ribosome assembled, small subunit and large subunit, clamping the messenger RNA between them. The E site is vacant because the tRNA that was previously involved has left. The P site is occupied by the tRNA carrying the growing polypeptide bound to its codon. The A site at this point is occupied by the tRNA bound to its codon that brings with it the next amino acid to be attached to the polypeptide. So let's now talk about the 3 stages of translation: initiation, elongation, and termination. Remember, I said these are the same stages we see in transcription, but very different things are going on in these 3 stages. So make sure you don't confuse the stages of transcription with the stages of translation. So initiation involves the assembly of the ribosome around the start codon and the binding of the tRNA to the start codon that brings with it the methionine that represents the first amino acid. So the small subunit uses the presence of the 5' cap and the 5' untranslated region, which is in here, to identify the AUG sitting here adjacent to that untranslated region as the start codon. So it then attaches to the messenger RNA, positioning itself with respect to the start codon. At the same time, the tRNA carrying the methionine binds to the start codon. This now allows the large subunit to position itself with respect to the small subunit and the tRNA, positioning that tRNA with its and the start codon in the P site, leaving the A site vacant, but sitting at the base of the A site is the next triplet of nucleotides, the next codon to be read in sequence. And so the next stage begins, elongation. Elongation is basically a cyclical process that continues so long as codons for amino acids move into the A site. So with the P site occupied by the growing polypeptide, the A site is vacant, but a codon sits there, coding for the next amino acid to be added to the polypeptide. The tRNA carrying that amino acid slides into the A site and binds to the codon via its anticodon. That triggers then the enzyme activity to break this covalent bond and form a new covalent bond, a peptide bond that links this amino acid to this amino acid, effectively transferring the polypeptide from this tRNA to this tRNA, but also adding in this amino acid and therefore elongating the polypeptide. That triggers the sideways movement of the ribosome, moving this tRNA into the E site and it exits, and moving this tRNA into the P site, which is appropriate because it's got the polypeptide in it. That vacates the A site, but also moves the next codon into the base of the A site so the cycle can continue. And this cyclic process continues until a stop codon slides into the base of the A site, which triggers termination. So with a stop codon, one of the three now occupying the base of the A site, a release factor, which is a specialized polypeptide, slides into the A site. That triggers the enzyme activity now to break the covalent bond linking the polypeptide to the tRNA, which releases it because there's nothing to attach it to. The tRNA now slides out of the ribosome via the E site because its job is done. With the release factor still occupying the A site, that triggers the two subunits of the ribosome to disengage from each other, which unclamps the messenger RNA, and translation is over. The two subunits then enter the pool of large and small subunits to at some point be reassembled into ribosomes, probably not with each other, but with other subunits to begin the process of translation all over again. Maybe on the same messenger RNA, maybe on different messenger RNAs, but the ribosomal units will continue to function as translation units. So there are a number of tricks that are carried out by cells, particularly prokaryotic cells, bacteria, that aid in increasing the rate of translation. So the first one, and to some degree, eukaryotic cells can do this as well, is to, rather than translate a single messenger RNA with a single ribosome at a time, to simply translate the single messenger RNA with multiple ribosomes. As soon as there is sufficient space for the binding of another messenger RNA to the 5' end of the messenger RNA, a new ribosome attaches to begin the process of translation. And that creates a structural unit called a polysome or a polyribosome. Polysome is the old name, polyribosome seems to be the preferred name now. So here you can see a polyribosome. This is actually in a bacterial cell, an E. coli cell. And you can see all of these ribosomes attached to the messenger RNA in this thread that you can see in here is the actual messenger RNA. Now, what this picture doesn't tell you, but what you'll see in a later illustration is that this is a messenger RNA that is still being translated, because up here is an RNA polymerase. This is actually a DNA thread. And so this is an RNA polymerase in the process of transcribing this messenger RNA. So in reality, in a bacterial cell, not only does the messenger RNA get translated by multiple ribosomes, translation actually begins as soon as the messenger RNA is long enough to accept the ribosome. In other words, transcription and translation occur in parallel. And that can happen in a bacterial cell because, remember, this is all taking place within the cytoplasm in a single compartment because there is no nucleus here. So transcription and translation can occur at the same time. And it is the presence of the polyribosome and what is referred to as a transcription translation complex that explains why bacteria can grow and divide so quickly. because they get on with the process of creating polypeptides from genes very, very quickly. As soon as the messenger RNA, and it really is a messenger RNA, the transcript is an intact, available messenger RNA, no introns to be edited out, translation begins immediately. And not just by a single ribosome, but by multiple ribosomes as the messenger RNA elongates, as transcription continues. A cool trick the bacteria can do. Polyribosomes can be seen in eukaryotes, not as frequently, but they do exist. But a transcription translation complex never exists because remember, two different compartments. So the other thing that we do have to take into account with eukaryotes is the fact that, as we've seen in the past, some proteins get excreted from cells, and that means that they need to enter the endomembrane system. And if that sounds a little bit fuzzy, you might want to go back and take a look at Chapter 6, where we discussed all of that. So how does the polypeptide get into the endomembrane system? Remembering that all ribosomes begin their life as free ribosomes in the cytoplasm, and that the ribosomes that give the rough endoplasmic reticulum its rough appearance come from that population of free ribosomes. Well, the answer lies in the gene sequence itself and the messenger RNA and the resulting polypeptide. As the polypeptide is assembled from the ribosome, the first few amino acids, and it could be depending on how far the polypeptide has to travel and how many membranes it has to cross, be somewhat lengthy. It can range from around 5 or 6 amino acids out to around 20 amino acids. But those first set of amino acids represent what is referred to as a signal polypeptide. That sequence of amino acids signals to the cell the final destination of this polypeptide. If it is to enter the endomembrane system, then the signal polypeptide is recognized by an SRP, a signal recognition particle. If I remember rightly, that's what the P stands for. This contains a small RNA molecule that in turn helps this particle attach to the signal polypeptide. This escorts the ribosome, the messenger RNA, and the elongating polypeptide down to the surface of the endoplasmic reticulum, where it binds to a translocation complex, a protein complex that acts as a gateway into the endoplasmic reticulum. The SRP binds to that and acts as a key to unlock the translocation complex. This allows the polypeptide now to be secreted into the lumen of the rough endoplasmic reticulum. At the same time, because the signal polypeptide has done its job, it gets cut off. So this would be a polypeptide that would not have a methionine as the first polypeptide, because it would be cut off as part of the signal peptide. The protein is now within the lumen of the endoplasmic reticulum. It will get packaged into a transport vesicle that will carry it to the Golgi, which will ultimately repackage it into another transport vesicle that will carry it onwards to the cell membrane, which will allow it to be secreted from the cell. In other words, to passage through the endomembrane system. But it all begins by the SRP recognizing the signal peptide and bringing the ribosome complex, including the peptide and the messenger RNA, down to the surface of the endoplasmic reticulum that allows the ribosome to anchor to the surface of the endoplasmic reticulum and secrete its polypeptide into the lumen of the endoplasmic reticulum, beginning the passage of that polypeptide through the endomembrane system. So this just reiterates what I told you earlier about the transcription translation complex in bacteria. No need to restate this. We've already talked about it, but here's a nice picture that just shows you. There's the polyribosome, DNA, and the RNA polymerase carrying out transcription. You can see that as the RNA gets longer, more and more ribosomes keep attaching to it. So this is the artist rendition of that. And of course, in eukaryotes, and this is the eukaryotic system, we know that we have to have RNA processing to remove the introns, splice the exons together. We need to do the capping and tailing. All of that needs to take place in the nucleus before the final messenger RNA is able to exit from the nucleus and then be translated by the ribosomes out in the cytoplasm. So that's a difference between prokaryotes and eukaryotes that you probably ought to be aware of. So let's stop again for a quick break, and I am going to break it here and come back after the break and talk about these concept check questions. So describe how a polypeptide can be secreted from a cell reaches the endomembrane system. We just talked about that, so you should be able to remember that. And then draw a simple diagram of a tRNA with the anticodon sequence 3' CGU to 5'. What are the two codons that combine to this? So obviously this may be one where the wobble base is involved. We may have to look at the genetic code to figure that out. Draw these codons, label the 5' and 3' ends, and identify the amino acid they code for. I think we're going to have to look at the genetic code for that. So I'll see you on the other side of the break with the answers to these questions.